The overall goal of this method is to increase solubility of hydrophobic compounds in aqueous solution for potential delivery into clinical settings.
This method addresses a key limitation in the medical field, which is the lack of use of hydrophobic compounds in clinical settings due to their poor solubility in aqueous systems such as the blood. The main advantage of this technique is that hydrophobic compounds can be dissolved in aqueous solutions without any significant use of toxic co-solvents.
First, label two 50 ml conical centrifuge tubes for each amino acid. Prepare a large 2-liter flask containing purified water. After calculating the amount of each amino acid for the desired concentrations, weigh the appropriate amounts of amino acids into their respective centrifuge tubes using a spatula. Using a serological pipette, add 40 ml of purified water to each centrifuge tube. After capping the tubes, vortex the samples until dissolved. Using a high-performance analytical balance, weigh approximately 1 ± 0.2 milligrams of peptide into the bottom of a 20 ml scintillation vial. Record the exact weight of the peptide on the cap and screw the cap on the vial. Next, pipette the appropriate volume of amino acid solution into each vial in order to reach the desired concentration of self-assembling peptide. Sonicate the samples for 10 minutes in a water bath sonicator at room temperature, ensuring the solutions in the vials are completely immersed in the water bath.